Welcome, everyone. So glad you're here. Today, we're diving straight into a true masterclass explainer on the foundational concepts of pharmacology. Now, whether you're a second-year medical student, an MD pharmacology resident, or gearing up for your NEET PG or next exams, this is really where your mastery begins. I'm going to guide you through this material today, but not just as a dry list of facts to memorize. Instead, we're going to look at the deeply logical, honestly fascinating narrative of how chemicals interact with human biology. By the time we wrap up this session, you aren't just going to know pharmacology, you're going to deeply understand it. Okay, let's jump right in. Here's a quick look at our roadmap for today. We're going to define what a drug actually is, trace the historical origins of the field, and then walk through the massive synthetic revolution of the 20th century. After that, we'll hit the modern biotech emergence, break down exactly how drugs act at a cellular level, and then we'll wrap it all up with a high-yield revision to lock it in for your exams. All right, starting off with Section 1, what exactly is a drug? Let's define our absolute basics. To really grasp this, I actually need you to abandon your everyday colloquial notions of what a drug is. As clinicians, we have to be incredibly precise. So read this definition with me. A drug is a chemical substance of known structure, other than a nutrient, which, when administered to a living organism, produces a biological effect. Now, pay close attention to that phrase, of known structure. That is a strict requirement in your standard textbook definitions. It's a huge umbrella. It covers synthetic chemicals crafted in a lab, purified plant extracts, and even intentionally administered endogenous hormones. For example, think about insulin. It's naturally produced by our pancreas, right? But the very moment you draw it into a syringe and administer it to treat a patient, it technically becomes a drug. Memorize this definition. It is literally the bedrock of everything we're going to do. And building on that definition, we get to this brilliantly classic high-yield viva concept. Back in the 16th century, the Swiss physician Paracelsus said, "All drugs are poisons. It is only the dose which makes a thing poison." This is absolutely crucial to understand. Even the most lethal toxins can serve as critical pharmacological tools in the right context. And conversely, everyday, completely normal medications can be fatal if the dose is wrong. This idea is the entire foundation of the therapeutic window, that safe little bridge between efficacy and toxicity. Think about paracetamol. A standard dose easily cures your headache, but a massive overdose, it completely destroys the liver. So always remember that poison-dose relationship. Moving on to Section 2, let's look at the origins of pharmacology, going from materia medica to rigorous science. You know, for centuries, humanity relied on a medical system that was, well, far from the rigorous biomedical science we practice today. We used a system called materia medica. This was essentially the dark age of medicine. It was heavily, almost entirely reliant on dogma rather than actual evidence. Believe it or not, even brilliant, legendary scientific minds like Robert Boyle back in the 17th century were recommending concoctions made of worms, dung, and moss scraped from a dead man's skull. Wild, right? Compare that to modern pharmacology. Today, we are strictly experimentation-based, relying only on purified, active chemical compounds. That shift from blindly following authority to rigorously testing hypotheses, that was the single most crucial turning point for actual patient care. To really build a mental map of this evolution, let's trace this timeline. The absolute pivotal moment came in 1805. A young German apothecary named Friedrich Serturner managed to purify morphine from opium. Now, why is this such a high-yield exam point? Because it proved once and for all that specific, purified chemicals, not magic, not invisible vital forces, are what actually cause biological effects. It completely opened the floodgates. By 1909, we had targeted treatments for syphilis. By 1935, we saw the very first antibacterial sulfonamides. And that momentum carried us all the way to the 1980s biotech boom and eventually the sequencing of the human genome in 2003. That brings us nicely to Section 3, the 20th century revolution, also known as the synthetic era. During the synthetic era, the fresh, innovative wind of synthetic organic chemistry completely revolutionized the pharmaceutical industry. We stopped just extracting things from plants, and we started actually building drugs, molecule by molecule. Paul Ehrlich's concept of magic bullets was absolutely monumental here. Ehrlich realized that a chemical could be engineered so that it was slightly more toxic to an invading microbe, or say a proliferating cancer cell, than it was to the patient's own healthy tissues. This concept, selective toxicity, laid the entire foundation for modern chemotherapy and antimicrobials. It's a brilliant conceptual leap, and one you must deeply understand for when you're integrating your pathology and pharmacology later on. Okay, Section 4, the biotech emergence. Welcome to the new era of therapeutic agents. So, since the 1980s, we've witnessed the dramatic rise of biopharmaceuticals. These are incredibly complex therapeutic agents, things like monoclonal antibodies, enzymes, and hormones. They aren't synthesized by chemists mixing things in glass flasks. Instead, they are produced by genetically engineered living cells. However, and listen closely because this is a massive point for your university exams, while their manufacturing process is entirely different from those synthetic small molecules we just talked about, their core pharmacological principles, the actual rules of how they act inside the human body, remain exactly the same. Which leads us to Section 5, how drugs act. Let's get down to the molecular mechanisms. We are going to return to Paul Ehrlich here for a golden rule of pharmacology, corporea non agut nisi fixata. Translated, that means a drug will not work unless it is bound. It's that simple. The drug molecule has to get close enough to a cellular constituent to physically and chemically interact with it. We call these cellular constituents drug targets. If there is no binding, there is absolutely no pharmacological response. It really is as simple and, honestly, as profound as that. Take a look at how these targets actually operate in the body. Within our physiological system, drug molecules have to physically dock into something, like receptors, enzymes, or ion channels. Once they are bound, they initiate signal transduction mechanisms, so maybe they activate an enzyme or maybe they pop open a channel. That action then cascades down to produce the ultimate biological response that you actually see at the end of the chain. This visualization right here, this is the absolute core of pharmacodynamics. To really reinforce that mechanism for your theory exams, I want you to lock these 4 fundamental targets into your memory right now. Nearly all drugs act on one of these 4 primary target proteins. A really great way to remember this for your vivas is the simple acronym RECI: receptors, enzymes, carriers, and ion channels. Seriously, whether you're dealing with a basic over-the-counter antacid or a highly complex modern biologic, it is almost certainly interacting with one of these 4 macromolecular structures. Now, this interaction relies on something called reciprocal specificity. Think of it like a highly specific lock and key. A specific drug fits only a specific target. But, and please pay attention to this because it is a classic MCQ trap, no drug is 100% specific. It just doesn't happen. As you increase the dose, the drug is inevitably going to start spilling over and binding to secondary targets. For example, a non-selective beta blocker that you prescribe to lower heart rate might spill over and hit lung receptors, which causes airway constriction. Clinically, this is exactly what manifests as side effects. This is Paracelsus's dose makes the poison, acting right at the microscopic molecular receptor level. All right, Section 6. It's time for our high-yield, last-minute revision. Let's review our critical takeaways. We're shifting into rapid revision mode now. I want you to take a mental screenshot of this quick reference table. You absolutely must know these historical titans for your exams. Paracelsus gave us the concept that the dose makes the poison. Sir Turner proved chemical causality by purifying morphine back in 1805. Ehrlich conceptualized targeted therapy with his magic bullets and arsenicals for syphilis. And Damach ushered in the whole antibacterial era with sulfonamides in 1935. Commit these 4 names and their discoveries to your long-term memory. Synthesizing our entire masterclass explainer today, here are your absolute non-negotiable critical takeaways. First, intent matters. Endogenous hormones are considered drugs the moment they're administered therapeutically. Second, scientific pharmacology truly began when we proved that purified chemicals, not magic, cause biological effects. Third, the 1980s biotech revolution fundamentally changed how we make drugs but not how they work. And finally, the golden rule. Drugs must bind to targets, remember RSCI, receptors, enzymes, carriers, or channels, to produce any effect at all. If you deeply understand these 4 bullet points, you possess a true conceptual mastery of this entire chapter. I'm going to leave you with a final, slightly provocative thought to chew on. If all drugs are technically poisons dictated by dose, and their action relies purely on binding to specific protein targets, how do you think the 2003 sequencing of the human genome is going to redefine the perfect dose for your unique individual cellular targets? The entire future of personalized medicine is literally being built right now on the exact concepts we just covered. Think about that. Keep studying hard, and I'll see you in our next explainer as we dive even deeper into the fascinating world of pharmacology.